started because I'm sure that Dr. Donovan has plenty to talk about, and we want to have some time for questions. For those of you who don't know me, I'm Barbara Fies. I'm the director of the Family Resiliency Center here. We are pleased that Dr. Donovan has agreed to present in the first of our Food and Family series. This is a new brown bag series that we initiated this year to highlight the research that is being supported by the Christopher Family Foundation Food and Family Program, and the intent of that program is to support innovative interdisciplinary research that highlights the positive connections between food and family. And so Dr. Donovan is one of our recipients in the SEED grant program this year, so I'll turn it over to her.
Okay, thanks, Barbara. So I'll be presenting, starting to describe a project that was recently funded, and then next month, Kelly Boss in the back will be sort of presenting on this as well, because as you can see, it's quite a broad project. This is the title, and the focus of my presentation is what is the potential role of the microbiome. So I'll be providing a brief introduction to the grant, and people who aren't familiar with Strong Kids, since this project is drawing from that, I'll just briefly mention that. What can we learn from poop and spit? We oftentimes ask ourselves this question, but we can actually learn a lot from poop and spit. So then I'll talk about the potential role of the microbiome, and some of this is we know a lot about from animal models, but there's much less known from human studies. And then tune in for next time. So both Dr. Bost and our collaborators and I responded to the Food and Family RFA. And we actually put in two different proposals. So one was gene-environment interactions effects on polymorphic variation of responsive feeding and picky eating with Natasha Cole and Jacqueline Saltzman as graduate students. And then Kelly put in this PI, one on self-regulation, childhood obesity with the gut microbiota. As you can see, the research teams were similar. There was quite a bit of overlap, and also the cohort that we were drawing from was the same, so Barbara in her wisdom said, why don't you guys put this together? So what we have is a big green grant now, which green for money, which is nice, but it includes all the research team members. But now we incorporate gene, microbiome, environmental interactions, and a multi-level effects on children's self-regulation, parental sponsored feeding, and family relationships around meal times. So the aims of the project are to identify associations between genetic makeup and picky eating behaviors at 12, 18, 24 months, to determine the effects of genetic composition, mainly single nucleotide polymorphisms, and observe parental feeding responsiveness at around 24 months. These are home observations between 18 and 24 months. And this is Natasha's dissertation, is really to define how nature or genetics interacts with nurture or the environment to influence feeding behaviors. We also will look at whether the early gut microbiome composition and temperament predict feeding behaviors, and we're hypothesizing that the microbiome is also influencing temperament. And then through family observations and meal times, look at the interactions related to children's eating behaviors, executive function, and weight trajectories. So you can see it's quite a broad proposal, very multidisciplinary, bringing one things we talk about in Strong Kids is cell to community, so we're bringing in the genetics and the microbiome with families and actual observations. So the Strong Kids program is a longitudinal cohort entitled the Cell Society for Nutrition in Early Childhood. We're recruiting women in the 3rd trimester of pregnancy. It's a birth cohort beginning with sample collection at a week of age and through 5 years of age. Our ultimate cohort will have 440 families, and it's funded by the Dairy Research Institute. The Strong Kids program, again, is one of the few that really aims to provide unique insights into how individual biology interacts with family environment. A lot of people in this space are either on the biology side or they're on the behavioral side. And very seldom do you see teams really coming together to bridge how biology, whether it be genetics or microbiome in this case, is potentially mediating some of these behavioral outcomes. So we're collecting a number of biological samples, which I'll show you on the next slide. We also have several sub-projects off of Strong Kids, and one is Protected Parents, which Kelly Boss is PI. And this involves home observations of the goal is 120 families of the Strong Kids cohort between 18 and 24 months. So the nice part about this is that we're actually going into the home, observing the family during meal times, looking at parent-child attachment, security, child executive functioning, family mealtime behaviors. So again, this is a lot of the areas where Dr. Voss has expertise and where Jackie Saltzman, the graduate student, is working on. So this outlines the Strong Kids. So you see we're collecting samples beginning at a week, 6 weeks, 3 months, 9 months, 12 months. We now have home observations between 18 and 24 months, and we've just been able to get funding to extend sample collection out to 5 years. So we have a number of components of a parent survey, which is quite extensive and ages up with the children, so we can look at developmental changes over time. So we're looking at nutrition practices and intake, daycare, family meal times, family physical activity, media exposure, sleep patterns, attachment. That's just part. There's actually even more that we're assessing. And then on the biological side, we're collecting height and weight from both the mom and child at all time points. Stool sample collections from all time points from the child and the mother at 6 weeks. We're getting saliva samples from the mother and the infant for DNA analysis. And we're collecting a milk sample at 6 weeks also. So I'm not as good at drawing these things as Kelly is. So I did my best shot at trying to show how we potential relationships between the measures. And so here's sort of the biology. So we have the child's genetics, we have the child's gut microbiome, and then the gut-brain axis, which I'll tell you a little bit more about later. These interacting to affect family meal times and picky eating behaviors, either directly through affecting child temperament, attachment, or executive functioning, or actually I guess I should have another arrow here, or mediating this effect. So this is where we're bringing in some of the questionnaire, survey, and observational data with the biology, but with the ultimate observations of family meal times and looking at picky eating behaviors. So this is very broad. Obviously, there's a lot of more specific hypothesis-driven questions within this. So just a brief survey of what can we learn from poop and spit. And just to start with, to say DNA from saliva yields informative SNPs. So this is work from Dr. Tran Garcia's lab, who's been driving this. So SNPs, if you think about them, these are single nucleotide polymorphisms. So these are a single nucleotide within the genome, within a gene in particular that's changed. And the modification is such that it's not going to cause such a detrimental effect that it would have been weeded out. So we have, I think, 67 million SNPs in the human genome, so there's a lot of them. We're learning a lot that some of these SNPs, because then they can encode for different proteins or amino acids and proteins, can affect disease outcomes. Either they can be protective or they can be cause risk of disease. So this is a lot of the work that Dr. Tran does, looking at different what we call risk alleles, or SNPs that have been associated with things like cardiovascular disease or cancer or eye diseases or diabetes or metabolic syndrome. You know, it goes on. So we have a couple, one paper published, one abstract, and the paper almost ready, and then another paper we got to review back, it's under revision, which have all looked at SNPs collected from, isolated from the saliva, the DNA and saliva of kids from Strong Kids 1, our first cohort. So one is looking at body weight, one is looking at picky eating behaviors, and this is again a more complex study that Dr. Boss will talk about, looking at how potentially a SNP in a gene is moderating the effect between parenting and parents' perception of BMI and feeding. Very, very interesting, very complex, but I think it's where the field is going. So I'm just going to give you a little taste of these two. And then again, as I'll show you later, there's quite a lot of emerging evidence that shows that changes or differences in the gut microbiome can affect weight, can affect behavior, and in adults can affect appetite and food intake. So this is the one study that has been published so far out of Dr. Tran's laboratory. So we chose 128 children from the Strong Kids cohort with a median age of about 3.5 when we studied them. This was basically taking a candidate gene approach. So we highlighted 8 different genetic variants in genes, which in adults have been shown that these SNPs have been associated with obesity or BMI or weight gain. So these were ones that we already had evidence that SNPs in these specific genes were associated with obesity. So then we looked at these SNPs, we related it to BMI and anthropometric traits using Z-scores, and basically our covariates were age, sex, and breastfeeding duration. And so this is basically the main point of what we found. So this is weight per age Z-score. So basically a Z-score of 0 is about 50th percentile. And this is a BMI percentile. So this is the numbers of SNPs that the children have. So in the genes that we looked at, they could have, we observed anywhere between 2 and 14 SNPs. And so this is this idea of this cumulative risk. So if you look at the bars, the numbers of SNPs was evenly distributed, and it showed like a nice bell-shaped curve. But then when we plotted on either BMI or weight per age Z-score, you saw that as children accumulate or had more of these SNPs, they tended to have a higher BMI. So this was saying that even in very young children, so these were a little over 3, that genetics is already playing a role. So even early in life, genetic predisposition is likely interacting with environment. One of the reasons actually we control for breastfeeding, because we did find that breastfeeding could help to moderate some of these effects. So it did show that interaction between environment and genetics. This was another study that Natasha Cole, who's in the audience, is the PI on, and this again was looking at a subset of samples of kids, 144 kids from the Strong Kids 1 cohort. Her research is on picky eating, and so the question on this is we had SNPs in 6 different taste perception genes, and the question that we said, are SNPs in these genes associated to parental perception of picky eating or their picky eating activities? So there's different SNPs, both umami, I can never say that right. This is kind of the savory taste. It's a relatively new taste, sweet, salty, sour, and bitter. And just to highlight what she found is that the children were reported to be picky by their parents. So 62% had a limited variety of foods, consumed a limited variety of foods. 60% reported to have food neophobia. 74% refused meats and vegetables, and 24% were overweight or obese, but we didn't find any association with weight and the SNPs. So of the different genes that she looked at, the one that we found associations with was gustin, and these were significantly different. So particularly, there were 2 SNPs in this gene that were associated positively with limited variety. specific preparations. So these were kids who wanted the food presented in a certain way and prepared in a certain way, or the parent perceiving them to be a picky eater. So again, really exploratory. We had the availability of the SNPs. We had thought the taste might have a role, but additional work is needed in a larger cohort, and this would be very nice to come back with actual sensory studies. And this is something, so Dr. Lee's in the audience, another one of the investigators who's a sensory scientist. So this idea is, do picky eaters actually have differences in taste preferences, and that's what's potentially leading to their pickiness. So just two examples of why we think it's worthwhile to look at SNPs. Why do we think that genetics might be playing an important role? And then Dr. Voss will discuss next time a little bit more specifically, even about another gene that we've been found to be related in this child-parent interaction. So now we're going to take a, so in the rest of the time, we're really talking about the microbiome. And some of you know more or less about this, so I thought I'd give a brief introduction. So when we talk about the microbiome, these are the microbes. So it's not just bacteria, it's fungi, yeast, viruses, archaea, protozoa that live in and on our body. So when we talk about the microbiome, we're talking about what's in our skin, what's in our mouth, what's in our ear, what's in your vagina if you're a woman. Your gut obviously is where most of them live. We find, so the microbiome, it gets a little confusing because people will refer to the microbiome as the organisms, but really the microbiome refers to the collective genomes of all of these the trillions of bacteria. And there's a little bit of controversy now in terms of, we heard for a long time that our bacteria or microbial cells outnumbered us 10 to 1, so we were like 10% human and 90% bacteria, but now people have kind of pulled away from that. But we do know that they outnumber the genes. So if we look at the collective genomes in our microbes versus the 22,000 genes that humans have, they are outnumbering us by at least 100 to 1. And so I always like to bring out the point that microbes were here before we were, right? So humans have evolved in a microbial world. So we oftentimes think about them as our minions, and we're going to take a probiotic and that's going to make us healthy, or we're going to treat this with an antibiotic and that's going to make us healthy, two ends of the spectrum. But the fact is that we co-evolved with them, and there's this sort of mutualism. So they actually do things for us and we do things for them. I mean, basically we give them a nice, comfortable place to live, and we give them food that they can eat, and we maintain this environment. But they also help to produce nutrients, particularly vitamins for us. They contribute to nutrient metabolism, things like fermenting our fiber sources that we can't digest into short-chain fatty acids. They have been shown to help calibrate metabolic function, this idea of being related to not only obesity and metabolic syndrome, insulin sensitivity. So these are all areas of research. Certainly educating the immune system being critically important in infancy. Maintaining community integrity. So if you think about, a lot of people look at the microbiota as an ecosystem, and the various microbes are filling a niche within that ecosystem. So the other thing you oftentimes hear is like, well, if I were to go and sequence out everyone in this room, we would all be a little bit different. And that's true. The deeper down we go, we're going to be different. You'll also see that even in relatively healthy populations, the composition of the microbiota can be different. So this has been, what do we call a healthy microbiota? We don't really know. We know in infancy a little bit better, but in adults we don't know. But when they look at the functions of what bacteria are doing, they're relatively constant, not only between people, but within different sites on the body. So what that's telling us is that various bacteria can fill the same sort of functional metabolic niche. So, we hear a lot about, you need to take lactobacillus, you need to take bifidobacteria. Those may not be the best probiotics. It's the ones that we have now, but hopefully in the future we'll find there may be even better ones that we can look at. So this idea of community integrity, and so when we say dysbiosis, that means that biome has been disrupted in some way. They also help defend against pathogens. There's a sort of a competitive exclusion, and there's a lot of interaction between the bacteria. And again, these microbial products and metabolites, so there's this whole area of metabolomics of these products that are made from the bacteria that either communicate with cells directly in the bloodstream or in the gut, but maybe even get into the bloodstream as well. So very important for us. What influences them? So I'm just going to go kind of quickly through the life cycle. One thing to think about is that when we look at variability among the individuals, gender, diet, age, hygiene, if you live in Bangladesh versus New York City, body site, antibiotic use, all of these things can affect across the life cycle. Okay. So if we look at birth, one of the main factors that affect the microbial community is route of delivery. If we look at early nutrition, early diet, so whether that baby's breastfed or formula fed or combined fed, whether that formula has a pre or probiotic added to it, when is our weaning foods added. Then as we look at sort of that 6 months to 3 years, family members and pets come in here, fathers as much, whereas early on it's much more the mother. Also the greater complexity of the diet. So it's thought now that by 2 to 3 years, kids have an adult type consortium. So they look fairly similar to adults. So then you say 3 years to adults. This is a very long period of time. There's some study going on now with teens, but we really need a lot more data in this area. So in the teens, we can see things that typically tends to be more stable, but can be affected by puberty, pregnancy, and menopause. And then the other area where we don't know enough yet is advancing age. So as we look at people in the older groups, what we start to see is the microbial species or diversity tends to decrease. Populations also they become more similar among individuals. So generally, early on, we're seeing increasing diversity, increasing numbers and types of bacteria, and then reduced diversity in the elderly. One thing to remember, so the Human Microbiome Project in the US, which informed a lot of our knowledge, was pretty much, 25 to 40-year-olds? So that was what informed the Human Microbiome Project. There were no babies, young children, and there was nobody older. And there's now papers coming out, and octogenarians, people over 100, so they're trying to fill in some of the blanks. So this is also a nice figure which brings in some of the other things that particularly affect the early development. So again, mode of nutrition, breastfed or formula fed, cesarean delivery, I'll show you some data on that. The genetics, so host genetics plays a role because we know that from twin studies. And so twins, identical twins tend to be more similar than fraternal twins or other siblings. But they're more similar than other children who aren't related to them. So, part of that's genetics, part of that's environment. Hospitalization, again, because oftentimes in the hospital people get antibiotics. So these things can go together. The environment, so when we look across countries, is that child born in a hospital, are they born at home? What does home look like in different places in the world? This is kind of an interesting area. Maternal infection and stress. And this has been shown primarily in animal models, but we're getting some data in human subjects as well. Gestational age of delivery. But again, a lot of times it's because these babies are cesarean derived and treated with antibiotics. So there's really not, I don't think there's good data to say that gestational age itself has a role. I think it's more how we treat preterm infants. So this shows a typical pattern in the genera or the genuses over the first 3 years of life. This is a relatively new paper. We used to think that bifidobacteria were really predominant, but again, with sequencing, we now know that there's other Bacteroideaceae are quite prevalent, and you'll see as we talk that is affected by route of delivery. So the idea to think about is that particularly in the first year of life, you can see a lot of changes. And then things tend to settle out a little bit. And then again, by 2 to 3 years of life, things seem to be fairly stable. So this is from a different paper, and the idea with this is they followed 98 mother-infant pairs, and I have a couple more slides from this, but they looked at them over the first year of life. So what they found is when they compared to the mothers who were purple, this shows sort of the similar, the diversity and similarity, so they seem to be moving closer over the first year of life. You can see this as well. This is birth, 4 months, 12 months, 1 year. This is called principal coordinates analysis. So you basically can see that infants at the same ages tend to cluster more closely together, but they're moving over time. And a lot of that is driven by changes in diet over those first 12 months. And then this, basically, we oftentimes talk about alpha diversity and beta diversity. Alpha diversity is diversity within a sample, and beta diversity is between samples. And so over that first year of life, we see that it's becoming, more complex, so within the sample, and less heterogeneous. So the babies tend to be sort of getting closer to each other. And you can see even by 12 months, at this level, they are, looking much more similar to their mothers. So this is this idea of maturation of the microbiome over the first year of life. And we know that this maturation is critically important to immune development, cognitive development, digestive development. So this is just showing what happens with cesareans. These are vaginally delivered. These are cesareans, so it's from that previous study, so they go out to 36 months of life. And the main thing to see here is that if you look at the, so actinobacteria is where bifidobacteria is. So when these babies are vaginally delivered, you can see early on that they already have bifidobacteria. If they're cesarean derived, there's a bit of a delay, but then it comes in. But the thing that's really flipped in these infants are the Bacteroides and the purple and the firmicutes. And so again, this is now the phyla level. So I call the phyla as like your 40,000-foot flyover of the microbiota, because within these 5 major phyla, there's literally thousands of different genus and species. If we look at adults, we're basically primarily firmicutes and Bacteroides. So infants are unique in that they tend to have more bifidobacteria. But there's a big flip. So you see that with cesarean derived, they have a lot more firmicutes, and if they're vaginally delivered, they have more Bacteroides. Why is that important? Work from Jeff Gordon, he was one of the first to show that in obesity, this is what we see. So in obese individuals, this ratio of firmicutes to Bacteroides is shifted. And firmicutes tend to have bacteria that are very good at scavenging nutrients from fermentation. And so not every study supported it, but there's just this concept that, we know from epidemiological data that cesarean-derived infants are more susceptible to obesity later in life. And this early microbial pattern might suggest that these changes could be seeded, pretty much right after birth. So this was another study from a very well-known group in Sweden. It's got a lot of press. When you have a sexy study, they do nice pictures for you like this and publish them in the journal. So this was the idea where they were looking at this interaction between route of delivery and breastfeeding. And they had just 98 mother-infant pairs. They looked at vaginally born breastfed, which is our gold standard, vaginally born bottle-fed, and then C-section. And so basically, what they could see from the very beginning, different bacteria that are colonizing the gut, and that some of these differences persist and are maintained. So the main, they found this interaction of route of delivery and nutrition. What was interesting is what happens at weaning. And a lot of people found, again, there's still some controversy here, that it was that introduction of solid foods that drove a change in the microbiome at 6 months of age, typically when infants get solids, but what they found in mothers who continued to breastfeed a lot, introducing solids didn't affect it quite as much. So it really seemed that it was the reduction of breastfeeding, not necessarily the introduction of solids, that was driving the shift in the microbiome that we see when solids come in. And they looked at signature species that were coming in at various times, again suggesting just non-random transitions, and that I'm not showing this data because it's just too much, but they looked at specific differences in nutrient and xenobiotics, so metabolism of strange compounds, foreign compounds, that were a mark of gut maturation of the gut microbiome. So it was one of the first studies that was able to follow these infants over time and really look at that interaction between the breastfeeding and route of delivery. And then I'm going to show just a couple on antibiotic use. And the first one is actually an interaction between nutrition and antibiotics, and the other is just antibiotics. So this is part of a longitudinal cohort, so it's only 198 samples. They're going to have like 3,000 kids. This is a trans-Canadian, funded by the Canadian government project, so it's the Child Project. They were particularly interested in intrapartum antibiotic exposures. And this is actually something we've been talking about with another project because, it's quite common, particularly with cesarean deliveries, that moms are treated with antibiotics. So the differences we see with cesarean is it because that baby didn't go through the birth canal, or is it because the mother was treated with an antibiotic? And because it's confounding, because pretty much all moms are treated with antibiotics, so it's going to be hard to tease that out. So they looked at that and route of delivery. They had breastfeeding reported by the mothers, and then they looked at the microbiota by sequencing the 16S ribosomal RNA, which is how most people do it these days. They don't culture bacteria like they used to. So what they found, and I'll show you just a few figures, that the use of this intrapartum antibiotics with either cesarean or vaginal delivery was associated with dysbiosis, but that breastfeeding can modify some of these effects. So, this was basically just looking at the diversity, and what they found that this, if we look just at 3 months, this was no breastfeeding, partial breastfeeding, or exclusive breastfeeding, that early on, the exclusively breastfed infants have a lower diversity. And this is likely because some of the components in breast milk, the oligosaccharides, are driving the development of specific bifidobacteria. But it actually sort of shifted, so not as strong, but at 12 months it was still significant, where the exclusively breastfed babies actually had more diversity. So this has been a dogma in microbiology that we usually say greater diversity is better, because if you have greater diversity, then it's less likely for pathogens to come in. There's just greater diversity. The thing where I would say that's not necessarily the truth is in infancy, when we know that some infants are highly colonized with bifidobacteria, sometimes even one species of bifidobacteria, and that's a very healthy microbiota. But why this is shifting, we don't really know yet, because it's an observational study, but there was a change. So I would say this actually, in early infancy, this is better. Later, they're following these infants for farther, through the first couple years of life at least, so we'll be able to see if these changes persist. They also, this is a little bit more complex, but again, we're going back to the phyla. So we talked already about, so the orange is the Bacteroidetes, Firmicutes, Proteobacteria. I guess it looked like orange to me, and then the Actinobacteria. So the main thing to look at, so looking at this site, so these are exclusively breastfed, vaginally delivered, no antibiotic, so we can say this is the gold standard. So if they're vaginally delivered with antibiotic, you can see that there's still a difference. So even if they're vaginally delivered, getting an antibiotic changed the relative distributions of these different phyla. And then whether the mother had an elective or an emergency C-section didn't really seem to differ if the mother breastfed. If we look over on this side, for non-exclusively breastfed at 3 months, this is different because, so these babies are likely being formula fed. And what we've shown in our own studies is that formula feeding is a very strong driver to make the microbiota be similar. So in this case, these infants, whether they had antibiotics or not, if they were vaginally delivered and formula fed, they look quite similar. But we did still see differences, particularly if we watch this group. So antibiotics with an emergency C-section versus an elective C-section, you can see that they're the most different. And I think the thing that we have to watch out for this is that they're just looking at the Bacteroideaceae. This is our group. This is at 12 months of age. So again, our gold standard group, seems like with breastfeeding, there's still diversity, but these were not significantly different. So this is this idea that breastfeeding may, in some ways, be able to compensate for the effects of early antibiotics in C-section. But the group, that was still different was this antibiotic with the emergency C-section. And so these differences were still persisting out at 12 months of age. And then looking at estradiolase, again, this is higher. So you can see that it was different than the other non-exclusively breastfed groups, and it was different from the gold standard vaginal no antibiotic, and then these weren't different. So this is this idea that while C-section and early antibiotics might cause this dysbiosis, if the mothers are breastfeeding, that these are no longer significantly different at 12 months of age. So this is really complex. Again, another really interesting study. They had like 39 infants, and they get into science translation of medicine. So these are babies who got a lot of antibiotics. Each of these little dots are different antibiotics over the first 3 years of life. These are babies who got like little or no antibiotics, and this is where they took the samples. So the nice thing about this paper is they looked at different classes of antibiotics, and what they found, and I'm only going to show one figure from this very nice paper, is that this is this idea that if you had no antibiotics, so this is they call it Jakarta index, so they're looking at how similar the samples are. So if it's purplish, that means it's highly similar, so 0 and 0 time points. So if you look at the same time points, 30 months and 30 months, they're similar. And then as the child ages, obviously the microbiota shifts, and that's why it's less similar. So even from the back of the room, you can see that the pattern differs between no antibiotic and antibiotic, where you see a lot more in the reds, the oranges, and the yellows. So there's much less stability in the microbiota for these infants who are treated with a lot of antibiotics, and particularly, around the times when they're treated with the antibiotics. So you cause this sort of acute disruption, but there's also this overall persistent disruption and lack of stability of the microbiota with continual, frequent antibiotics. And this just shows, again, much higher numbers, which is less stability, so that there's more variance. So antibiotics, and now we know early antibiotics are associated with so many diseases that have been associated with the microbiome. So things like obesity, autism, other diseases. The issue is, before I kind of get into this part, where we are with the microbiome research and why I'm saying potential here, because I think we have I should be starting, here we are, this last part. A lot of where we are is cross-sectional studies. A lot of it is what is referred to as phenomenology, where they say, oh, this person is obese, let's look at their microbiota. And this person has autism, let's look at their microbiota. So it's associative. There's very few studies in humans that are actually being able to show cause and effect, because we just can't do that. So most of our evidence that's mechanistic and cause and effect comes from animal models. So this is a small font, but it sort of sets out this idea of the gut microbiome and the gut-brain axis. And then I'm going to show you some pretty pictures. But The other thing that's emerged is this idea of this prenatal, susceptibility to changes in neurodevelopment, which I think people have understood. But we now understand there's actually a microbiome that the baby's exposed to prenatally as well. There's microbes in the placenta and the amniotic fluid. They swallow those, and so when they poop out their first meconium, there's already microbes in there. And so, in some studies out of Finland have shown that potentially these microbes are even already interacting with that fetus's immune system and can be different if the mom takes a probiotic, for example, during pregnancy. So one of the areas that people have really started to look at is looking at how these neurodevelopmental pathways and how the microbiome is changing over time. So, ideally, in the old days, everybody vaginally delivered and breastfed. So through most of human evolution, that's what the brain and the microbes were exposed to. We now are able to have cesarean sections and antibiotics and lots of other interventions. So exposure to adverse events that interfere with this critical sequence of development has a high risk of subsequent emergence of mental diseases, illness later in life. So there's a lot of people who are really looking at early life antecedents of mental illness. Again, as we talked about, several studies have shown that early life events can impact the gut community. So what's the interaction? Other lines of evidence show there's this bidirectional communication between the gut and the brain. I mean, most people have probably known, like when you get nervous, you feel it in your gut, but your gut, we now know, actually communicates to your brain as well. So there's a bidirectional interaction. Again, the gold standard trajectory of our gut microbiota, the breastfed, vaginally delivered infant, that deviations from that can lead to not only disorders in the gastrointestinal tract, but complex and metabolic and immune disorders. So I showed you some of the things that can push that trajectory off. Okay, and so now these studies are being extended to other disorders of the central nervous system. Pretty much everybody is doing microbiome work these days. So this is just basically another very complex figure, but this is our current understanding of sort of the gut-brain axis. And what's been added is the microbes. So for a long time, there's this understanding of the interaction between the autonomic and enteric nervous systems, the neuroimmune system, and the neuroendocrine systems. So the gut, for example, our microbes make tryptophan, and which can be converted to serotonin. And so what Kirsten is doing with autism suggests that this might be a way that dysbiosis can be linked to autism. We see stress hormones, so this is where when you're under stress, things may come in. You see immune cells and cytokines. These are all being influenced by the microbiota. We now know, like I said, that the microbes themselves make neurotransmitters as well as tryptophan, and they create these short-chain fatty acids from fiber fermentation, which can interact with immune cells, and there's even receptors on our nerves for them. Okay, so a very complex interaction that includes the immune system, the endocrine system, and the nervous system. And just to remember that it's bidirectional. So your gut informs your brain, and your brain informs your gut. So this, just to give you a picture, so I said a lot of the studies, a lot of the data that we have is really from animal models. So how have people looked at the microbiome gut axis? They've looked at it in germ-free animals. Probably some of our best evidence is from rodents that we can raise completely germ-free, and that's where you really see that they don't develop normal cognitive and behavioral development. Infection studies, so areas where you can disrupt the microbiota. Some human studies with probiotics, things like yogurts and antibiotics have been done. Fecal transplantation, so they can take a stool sample from a human, put it into a germ-free animal, make it notobiotic, and look at the behavior. And so, this, if you're interested in this, I'd say this is a really great review. John Crine's a leader in this area, and so that he reviews a lot of the studies. This is this idea, again, going back to, what's happening with the microbiota, increased complexity, what's happening to the brain pre and postnatally. So myelination, there's just recently a paper showing that the gut microbiota can influence brain myelination. There's work also with synaptic pruning, which is critically important. So this was a paper actually from the same group in Cork, Ireland. And, they basically said there wasn't prenatal, possibilities for interventions or modulation, but this is another recent review where they were really looking at the different diseases. So what they were putting together really nicely was the microbiota, the neuronal complexity. So you can see in infancy it becomes more complex, and then we prune, and then, unfortunately as we get older, we're getting less complex. But, they were, feel it every day. So they're basically looking at these relationships between the microbiota, what's happening in the brain, and age of onset of different mental disorders. And so again, they're really looking at, much more from a disease perspective. So also from the same paper, but again, they really brought in, okay, where are our potential windows of opportunity? So they are looking at prenatal through pre or probiotic supplementation. Again, mothers who are consuming certain probiotics, you can find some of the same in placenta, amniotic fluid, and in the infant's meconium. And then after, again, through breastfeeding, potentially through diet, breastfeeding or formula, and then this idea of the gut, being able to feed back and modulate the brain. So while there's not very many papers in children, I thought this was probably the most appropriate for the type of work that we're going to do. So this was a paper from a group out of Ohio State looking at gut microbiome composition and temperament in early childhood. So they had 77 children, 41 boys, 36 girls. They were on average about 2 years of age, primarily Caucasian. The kids were excluded if they had any major health condition or developmental delay, or if they were actually toilet trained, which I thought was interesting because they thought it hindered stool sample collections. So, basic method, 16S ribosomal RNA sequencing. They used the ECBQ questionnaire for temperament. They asked the mothers how long they breastfed, if they breastfed, when formula was introduced, age at which solids were introduced, and the current frequency of intake of their kids. So they had a little bit of nutrition data. So they did see temperament differences between boys and girls, and I think probably there's people more expert in this room than I, but they basically found that there were sex differences in 4 of the 18 scales on the measure. Boys had higher ratings for motor activation and high intensity pleasure, and those, as you'll see, were actually related to the microbiome. Girls rated higher for having inhibitory control and suitability. They found that significant differences, again, were when these are scales that can be made into a composite. So we're going to see this surgency in extroversion. Boys were rated more highly on this, the squirrely little boy, and that was related to microbiome. And girls were more highly in effort for control. So what they found is that they didn't see gender differences. So when they looked overall at the microbiota of girls and boys, the overall community structure didn't differ by gender. But then as they started to break it down, they started to see gender differences. So this is looking at overall diversity. And so this is with surgency, extroversion, sociability, and high intensity pleasure. So these are 2 subscales of this. And in this case, they found that with both boys and girls, that as their phylogenetic diversity, as there were more diverse microbiota, the scales increased. Only in boys did they see this relationship with sociability and high intensity pleasure, not in girls. So they did see relationships with overall diversity in the microbiota. This is a little bit hard to see because this is actually kind of in a 3D structure. But what then they did is they broke it down to the different scales. So in this case, sociability went from 3.75 to 7. And what they found, again, that they were in these subscales, there was significantly different association with diversity. So they were able to pull this out. And this again was just in the boys. So again, looking at not only overall diversity, but there seemed to be different clusters based on how they were scoring along these different scales. So again, boys, sociability, high intensity pleasure, and activity. I see I'm running a little late, but I don't have too much more. So for girls, the only thing that was significant was fear. And so they found a relationship, which is part of the composite scale of negative affectivity, was the only thing that they were able to see differences along the scale of fear. So these are, again, looking at the bigger picture. Then they went down and said, well, can we actually attribute this to the relative abundance of any particular genus? And so what they found for boys, that there were 20 genera that accounted for about 92% of sequences, and for girls there were about 18. So for boys, there was significant positive association with sociability, with Chromococci, Parabacteroides, and then for high intensity pleasure and activity level, Dialster and Recanalsali. So when you do this, you have to learn how to say these names. But this is actually one that was interesting because it was related to high intensity pleasure activity level, but also the fear measure in females. So I was trying to do a little bit of reading on this and to try to figure out what's known about this. Sometimes the issue we have with sequencing is that you identify a lot of genera or families that we don't really know what their functions are yet. So it's really more identifying it. But then, what you can do is you can take this, go back into a germ-free rodent model to test your mechanisms of action. So, but the bottom line, they were showing overall diversity related to these scales, and they were able to show positive associations with specific bacterial genera. They didn't really see too much with diet. So with boys, the temperament characteristics that were significant previously, there was no effective duration of breastfeeding, age of introduction, but they did find that high intensity pleasure was associated with lower frequency of meat and vegetable consumption, so maybe a marker for a less healthy diet. For females, there really wasn't any association with breastfeeding solids, and they looked at fear and effort for control, and again, there was no association with current intake. Again, it really wasn't a very robust measure of intake. But again, to me, I think this is maybe more of a marker of less diversity in the diet or a less healthy diet than these foods actually doing anything particular. So again, the only study out there right now on the microbiome and temperament in children, I've kind of already mentioned most of these other. So gender-specific effects were observed with temperament and the relationships between the microbiome and these temperament, differences in temperament that were observed. There were some differences with dietary patterns, but they don't think that this didn't account for the observed difference in the microbiome. So limitations against a cross-sectional observational, but it does suggest that some of these bidirectional, gut-brain relationships, may be important early on. And again, the strength that we have in the strong kids, because it's a longitudinal cohort, is we'll have much more detailed information about early nutrition, current nutrition, and all the other life factors that go in. So again, just summarizing, I hope you feel now that the gut microbiome is essential for human health, plays a role in major bidirectional communication with the gut and the brain. The microbiome undergoes this vigorous process of development. Again, genetics, but then how that baby's born and how they're fed can either help that or disrupt it. Then the data that's coming out from epidemiological and some basic studies show that disruptives or perturbations can have short-term and potentially long-term adverse effects. So again, most of the studies are cross-sectional, therefore strong kids longitudinal cohort I think will provide critically needed data on these factors influencing development. And specifically the Food and Family Grant will enable us to examine, I think much more robustly, the associations between infant genetics, microbiome, temperament, and feeding behaviors. So just a plug for Kelly. So if you come back on the 19th, she will kind of talk about the same project with more of a focus on the gene-environment interaction, but we'll be sort of building on the same one. And I know we're running a little late, but if you have questions, I'd be happy to take any. Everybody's hair is blue.
So this is just kind of a general question. So cesarean section versus vaginal delivery versus breastfeeding versus formula fed. Maybe breastfeeding is more optional, but cesarean versus vaginal is quite not optional in most cases. So seeing the adverse effect in the microbiome, has any of the papers discussed what are some interventions or remedies?
Well, actually, there's a cool studies out now. Maria Dominguez-Bella, who's in New York City, has started a clinical trial in actually in Puerto Rico where the incidence of cesarean delivery is very high. And this paper's been published recently. So these are women. So there is a difference. So if you saw that one paper, and what people are starting to really look at is elective versus cesarean. Because some people could say, well, elective cesarean is voluntary, you know, because a lot of times it's done just because you had one the first time. Where we really see the differences seem to be with the emergency C-sections, and that is women who've undergone some degree of labor typically before. And this has even been shown in the milk microbiome, and it's thought that some of that early, the labor may be disrupting barrier function in the gut, and that's why you see some translocation of bacteria into the maternal blood supply, which then influence the milk microbiome. So I got off topic. So what Maria has done is women who come in for the elective C-section, she basically puts a sterile cloth in the birth canal for a couple of hours, an hour, it's not very long, while they're in the hospital waiting to have their C-section. And then after that baby's delivered, she takes that cloth out and swabs the baby down. And they're seeing some benefit from that. But again, I need to get it in the baby's mouth. But it's not gross. I mean, this is the thing. But you know, also, when the baby is delivered, it's not only the vaginal microbiome, they're also, you know, the fecal microbiome, because it's not sterile down there, right? And many women, you know, it's kind of rough, so things happen. And if you think about that's evolution, I mean, that's how babies got colonized. There's all these great, like, koala mommies give their babies poop, because they know when they have to go off breast milk and they have to start eating, you know, eucalyptus, they need their mommy's microbiota, so the koala baby mommies know they just feed their baby poop at a certain stage, and then they can start eating trees instead of mother's milk. So, you know, it's not, vaginal microbiota is not gross, human milk is not gross. People tell me all the time human milk is gross, you know, I never drink human milk, it's gross, but I mean, that's how we've evolved to have these inocula at a really critical period of time, and we've short-circuited that, you know, through the way we deliver babies. And even babies who are vaginally delivered, it's a pretty sterile environment, you know, in a hospital. And then how we treat them. And we're getting better about that, you know. I think women are, because of the hygiene hypothesis, which suggests that we were actually too clean with babies. So, you know, we didn't need to boil everything before you use it on your infant. And some microbial exposure is important to get that immune system to develop. So what Maria is doing is a first step. I think there's work going on, and I would imagine, you know, within the next 5 years there will probably be a probiotic inoculum for cesarean-directed infants. I know somebody's working on it. And, so if you're cesarean drive, a squirt of good healthy bacteria, and it's gonna make your microbes look more like a vaginally delivered. So when you see that come on the market, buy stock. In the U.S. and in our Strong Kids cohort, I think we have about 28% cesarean, but there are places in the world right now, the areas of Brazil, but even places like China, places in India, where cesarean rates are 90%.
What?
Yes. And so there's this huge experiment going on of what are going to be the health outcomes. Because also, particularly like in China, there's very little exclusive breastfeeding. It seems to be changing, but so you have this high rate of cesarean and then partial or no breastfeeding. So they're basically moving infants to the worst.
I'm getting called from Texas.
I'll decline that. Okay, any other questions?